address seluruh peserta yang mungkin juga tidak seluruhnya berasal dari Indonesia. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable Chairwoman of Indonesian Society for Biochemistry and Molecular Biology, or PBBMI, Professor Rahmawati Ridwan. Thank you for being here today, Prof, and giving us the opening remarks. Honorable Head of Department of Biochemistry, Faculty of Medicine, University of Riau, Dr. Eni Carmila Asni. We are so grateful for the faculty endless support for this webinar. Honorable keynote speaker, Dr. Riza Arif Putranto from Indonesian Biotechnology and Industry Research Center, and of course, Dr. Hanan Hasnan, our guest from Food Science and Technology University Putra, Malaysia. Thank you for your time and for your willingness to share valuable knowledge in CRISPR technology. Respected members of PBBMI, dear colleagues, and all participants of PBBMI webinar series. First of all, it gives us immense pleasure in welcoming all of you to the webinar series of PBBMI with the topic on CRISPR and genomic technology. CRISPR itself is an emerging technique in biotechnology, which holds promising development in genomic research. Moreover, with latest issue in COVID-19 pandemic outbreak throughout the world, we truly need fast but also accurate breakthrough in early detection and in biomolecular engineering design. And Alhamdulillah, this webinar has attained astonishing appreciation with more than 300 participants from Indonesia and neighboring countries. Inshallah, such webinar will be part of our regular PBMI program, because on previous sessions, PBMI Pekanbaru held a successful hands-on workshop on using RT-PCR and bioinformatic tool for supporting biomolecular and protein engineering research. And PBBMI is also committed to upgrade its member capability and capacity, also always ready to contribute to the community with new update of technologies in biochemistry and molecular biology. We are extremely hopeful that this webinar will be successful and all the participants can reach their goal in understanding the new technique in CRISPR technology. Lastly, we would like to thank our participants for joining our association, and inshallah we will follow up your membership as soon as possible after this webinar by sending registration form and directing you to the nearest PBMI branch in your city. Thank you for all your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Dimas. Ladies and gentlemen, next we will hear a few words from the head of Biochemistry Department of the Faculty of Medicine, Universitas Riau, to Dr. Eni Carmila Asni. Time is yours.
Thank you, Mr. Ahmad. Assalamualaikum warahmatullahi wabarakatuh. Good morning for all our distinguished guests. Dr. Emma, as a head of ESBNB, and also for Pak Dimas, and for head of SBNB Pekanbaru chapter, and also for our distinguished speaker, Mr. Riza Putranto and Mr. Hanan Hassan. Morning all, and also good morning for all college, ladies and gentlemen. Thank you for joining this webinar. On behalf of our Medical Faculty of Riau University, let me say that how honored we are to collaborate with PBMI Pekanbaru chapter in organizing this webinar. CRISPR-Cas9, which is engineered by Jennifer Doudna and Emmanuel Carpentier, enable us to cut DNA more precisely than other technique. This, however, also opens a new window for unimagined capability of genome editing. Sometimes I think that are we playing with gut capability of edit our genome? Because it had been used for a wide range of organisms, from yeast to human embryos. Of course, this technique also brings us to a long debate of ethical issues. Today, fortunately, and we have our distinguished speaker, Dr. Riza Arif Putranto, DEA, and Mr. Hanan Hassan, PhD, sharing their knowledge and experience in CRISPR-Cas using system. I do hope we could learn and share with each other about CRISPR-Cas system, and there is an open chance for us to do more collaborative learning and research in this field. For closing this opening remarks, please let me have one pantun session. It's usually some habits in Malay tradition. I will use Indonesian language for this pantun. Cegah COVID-19 dengan pakai masker, jaga dengan CRISPR untuk menjawab tantangan di masa depan. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Nicely done with the pantun, Dr. Eni. And now, ladies and gentlemen, let's hear an opening remarks and to officially open our seminar from the chairperson of the National ISBMB to Dr. Rahmawati Ridwan, or Dr. Emma for short. Time is yours. I'm really sorry, Dr. Emma, we cannot hear your voice yet. It might be the earphone is not compatible. I'm sorry, no, still no sound from your side. Try to unplug the earphone maybe, ma'am. Unfortunately, not yet. Ladies and gentlemen, while we are waiting for Dr. Rahmawati Ridwan, Dr. Emma, for the technical difficulty, allow me to introduce herself for a moment. She is the chairperson of our national ISBMB, the Indonesian Society of Biochemistry and Molecular Biology, or for in Indonesian, PBBMI Pusat. I believe it is her second round as the chairperson. And well, we have done a lot during her presidency, and one of them is establishing the journal from our society. We already have a journal called Acta Biochimica Indonesiana, and we hope that not only members, but all participants here can publish their research articles in the journal. Now back to Dr. Emma. Let me try to, no, we haven't been able to hear you. I'm really sorry, Dr. Emma, we are still not able to hear you. Maybe we can move your session into the closing remarks after the discussion session, if it's okay with you. And we hope that we will be able to overcome technical difficulties now. I'm really sorry, thank you. Okay, ladies and gentlemen, we will hear back from Dr. Emma in the closing session, so do not leave it after the discussion. We still have many things to listen and a photo group session afterwards. But now let's move forward to the most awaited sessions, the presentations followed by discussion session that will be guided by our moderator, Ms. Yuli Haryani, or Ms. Rani for short. Ms. Rani is a PBBMI Pekanbaru chapter member. She's also a lecturer in Department of Chemistry, Faculty of Mathematics and Natural Sciences of the Universitas Riau. And she is currently a PhD candidate in University Putra, Malaysia. Ms. Rani, time is yours when you're ready.
Thank you, Mr. Rahmat. Can you hear my voice?
Yeah, clear. And
Assalamualaikum and good morning all participants. My name is Rani Yuli Haryani. Thank you for joining us. It's my pleasure to be a moderator for this webinar on CRISPR in genomic technology. We will have an hour lecture. It will be a parallel session of 30 minutes presentation from the first speaker and will be followed by another 30 minutes presentation from the second speaker. Kepada semua peserta, sesi kali ini kita akan sesi paralel untuk kedua pembicara, diawali 30 menit untuk pembicara pertama, Dr. Riza, dan kemudian dilanjutkan 30 menit dari pembicara yang kedua, lalu dilanjutkan dengan sesi tanya jawab. Now we can begin. Our first speaker is Dr. Riza Arif Putranto. Dr. Riza Arif Putranto is a scientist expert in plant physiology using molecular biology approach, as well as bioinformatics, especially on NGS and protein modeling. His actual research interests focus on understanding cellular and molecular mechanisms during latex harvesting, developing CRISPR-Cas9 methodology in oil palm, and developing plant-derived products using in silico modeling and synthetic biology. He holds specific expertise such as genomic, transcriptomic, as well as metagenomic analysis. He is actively involved in collaborative research between France and Indonesia, collaborative network of bioinformatics with Informatics Center of LIPI, I3L, as well as other partners since 2015, and excellent partners of IoT technology with IPB University and boards since 2019. He graduated with a PhD degree in 2014, and he's now the head of research division at PT Riset Perkebunan Nusantara, Indonesia. And today, he will be sharing with us his expert experience on CRISPR. Without any further ado, please give a warm welcome to the first speaker, Dr. Riza Arif Putranto.
Thank you, Ms. Rani. Thank you for your warm welcome. First of all, I would like to thanks to the PBBME for these invitations, and without in honor to Mr. Hanan Hassan, PhD, I will give the speech in English if you don't mind, so that we can discuss clearly and without any translation between the two languages. So today I would like to present to you about CRISPR, and let me share my screen first. Okay, this one. Okay, thank you very much for this opportunity. Actually, I was so honored receiving the invitation from PBBME, and I was glad also that I can maybe share my point of view and also our research results in Bogor, in our laboratory in the Indonesian Research Institute for Biotechnology and Bioindustry. This research has been going on for 5 years, since 2015, and now we are reaching the end of the experiment, and we are hoping for the best result that we can have. So this morning, through this opportunity, I would like to present to you about CRISPR in general, and also I would like to share our knowledge and our experience in doing CRISPR, especially in oil palm in agriculture, because my institute works on the plantations, and Pekanbaru Riau as well is one of the biggest province in Indonesia that's planting the oil palm. So the talk with this morning entitled "The Breaking Wall of CRISPR." So I will skip my CV because Ms. Rani has already informed you about it. And this is the outlines of my presentations. I group my headlines of the presentation into 4 themes. First one, I will talk to you about the philosophy of CRISPR. Maybe some of you already know the technology, but some of you maybe don't. So I would like to try to give a more in general perspective about the CRISPR. And then, of course, the introduction of CRISPR and the research itself. I called the research as the breaking walls of Ganoderma, because we are trying to overcome the problems of Ganoderma in oil palm plantations. And I will end my presentations with giving you the facts and the complexities of CRISPR. Because sometimes when we talk about the technology, we always talk about the benefits, and we never talk about the difficulties or how we can apprehend the problems. So I will start to you with this. So maybe some of you are already seen this video or already known about this presentation from Steve Jobs in 2007. I would like to introduce you about CRISPR using iPhone, meaning that CRISPR nowadays is similar to the first iPhone that being introduced to the public many years ago. If you have seen the video, maybe you can imagine that the audience that coming to the presentation of Steve Jobs is owing. He's always say, it's very good, it's very nice. The technology looks very modern, etc. But it was 2007. Nowadays, if you see the video, maybe you see it's normal, it's nothing new, etc. Why? Because the technology always moving forward, and CRISPR now is something that is really new for us, and we can move forward after maybe in 10 or 15 or 20 years from now, we will see CRISPR as a usual technology. So if you can imagine, is there any CRISPR edited fruits or vegetables already in the market? And the answer is yes, because we are already worrying about the GMO for the past 20, 25 years. And now we have CRISPR, people worrying also how if this technology is even worse than the previous one, how if we cannot detect the technology or the plants that edited or similar to the GMO. But eventually the USDA or the US Department of Agriculture has granted approval for many fruit and vegetables edited by CRISPR. One of the example I showed you here is from the next gen plants of Australia. They have been granted approval to conduct field trials on their gene edited virus resistant tomato lines. They have 6 tomato lines resistant to the tomato spotted wilt virus and cauliflower mosaic virus, and developed at the University of Queensland. And the USDA has determined that these 6 tomato lines do not fall under the federal regulations of a GMO, meaning that it's not transgenic. And meanwhile, in many companies in the world, such as Corteva, Pairwise, Tropic Biosciences, Yield 10 Biosciences, they have also developed their own fruit and vegetables, such as waxy corn. You have corn that is really nice, nicely formed with a good productivity, etc. And then you have this soybean and also corn from the Pairwise that is in good quality, high resistance of disease. You have coffee from Tropic Bioscience, and you have camelina to produce oil, etc. So you have many fruit and vegetables coming out in the market, but unfortunately we have seen not yet one in Indonesia. I don't know in Malaysia, but in Indonesia we haven't seen one. But maybe in the forthcoming years we will also able to find this product in the market. But meaning that this technology is already common in the other part of the world. So we are now inside a new CRISPR and Cas world. I will try to give a more simple explanation about this. If you go to PubMed and you type genome editing or you type CRISPR, you can find many publications from high impact factor journals until you have notes also. And nowadays in 2020, you have more than 24,000 publications containing the keywords of genome editing and CRISPR. CRISPR technology, they say it's a simple and yet powerful tools for genome editing. And also it allows you to easily alter DNA sequence. That is the goal, alter DNA sequence, modify gene function. And in the aspect of medic, we also use the CRISPR and Cas technology to correct genetic defects, to treat and prevent spread of the disease. And now maybe some of you know that CRISPR and Cas has already been used to detect the virus of SARS-CoV-2 in COVID-19. And the team in Fengzhang in United States is already successful about this, and they have already published and also already used the product. So CRISPR is changed the way we do genetic science actually. So if you already know also that CRISPR and Cas patents or belongs to Jennifer Doudna from the UC Berkeley, Emmanuel Charpentier is a French lady from CRISPR Therapeutics, and also Feng Zhang from Broad Institute. They are competing one of each other. They build their own empire here in the circle on the right. They have their own industry. They have their own companies working on CRISPR and Cas. Before it was in therapeutics, in humans, now they are going also in agriculture, and even more recently they went to the COVID-19 problems. So this CRISPR wars has already changed everything about the genetic modification. But if you come back to the CRISPR, and if you go directly to the agriculture aspect, CRISPR is actually only, yeah, I said only, one of the new genetic modification techniques. We can summarize from the paper of Seiran and et al. In 2018. They have shown some technologies that exist today. They call it NGMT, a new genetic modification technique. As you can see here, CRISPR and Cas9 is only one of them. And we have this scope of genetic change. The more you have change in the genetic, going to upside, and you have more of genetic material combinations go to the right. So meaning that you have this current technology of transgenesis. You have recombination, protein recombination, gene recombinations, grafting, cisgenesis, agroinfiltrations. Everything falls into this kind of previous technology that we still use today. They use many DNA insertions, so greater DNA insertion and greater combinations and greater risks also. But CRISPR and Cas9 and other technologies such as TALEN, zinc finger, ODM, and also site-directed nuclease 1, 2, and 3. This is the type of SDN. They change as little as possible, the sequence by repair. And then as you will have also change of genetic material, not too wide, but directed, the one that you wanted. So that is actually the goal of CRISPR as a new genetic modification technique. Like when you have in the medic or the Faculty of Medicine, also in the medicine world now, you have what we call laparoscopy. Is like a minimized surgery, so that you can have a change, but you do not destruct many cells or many part of the human bodies. It's actually the same approach when you're using CRISPR. So, but maybe some of you were asking, actually, do you know where the CRISPR technology comes from? Maybe they say this was invented by humans by trying to make different cloning, etc. But actually, we learned it from the nature. Maybe you already know about the war again between the bacteria and the bacteriophage. So bacteria is the one that is very big here in the brown color. Bacteriophage is the virus that infects bacteria. So actually, it was a never-ending adaptive immunity of bacteria against virus, virus against bacteria, bacteria against virus, and go on and on and on. For example, when you have the virus infecting the host cell, just like COVID-19, they release the genetic material of the bacteriophage. It's a DNA phage. So the DNA recognized by the bacteria and being cut using a system, what we call a restriction enzyme from the bacteria. The virus recognized the restriction enzyme from the bacteria and try to modify and adapt with it. So they create a methylations on the genome so that the restriction enzyme cannot recognize the DNA phage from the bacteria. The DNA phage from the bacteriophage, from the virus. Bacteria can be infected again, but the bacteria did not stand still. They created a new restriction enzyme and cut it again and go on and on and on. And they're always doing something new. And one of these evolutions that the bacteria did, it was creating CRISPR. So what is CRISPR actually? CRISPR stands for from a clustered regularly interspaced short palindromic repeat / Cas-space associated proteins very long. Right? If you can remember it, very good. But if you don't, do not go hard on yourself. So why we call it clustered regularly interspaced short palindromic repeats? Why? Because we have cluster. So we have this cluster of sequence regularly interspaced. So it's separated by spacer. The sequence that have no meanings is like a fence, you know. And then you have short palindromic repeats. It's like a sequence that is palindromic, it's repeats. Like when you see on the left here, the color in red, in green, and also in purple, it was the CRISPR. The CRISPR itself, or the palindromic repeats, that is the red color here, comes from the virus, been recognized by the bacteria and been saved in the genome of the bacteria as an immunity system. Because CRISPR is actually the original defense system of the bacteria, for example, Streptococcus and Neisseria. So they recognize the sequence of the virus, and if any future attack from the virus, so the bacteria can recognize it and can cut the DNA sequence from the virus, and then the virus cannot infect the cell of the bacteria, and the bacteria survive. And CRISPR and Cas9, especially here, is only one of the type of the defense system. We have many classes. We have many types. We have two classes of CRISPR and Cas immunity system in bacteria. We have class 1 and class 2, separated by type 1, type 3, type 2, type 5. The one that separated the type and also the class is actually the protein, what we call Cas. We have from Cas1 to Cas13a right now. We have so many, but maybe more to find. And we separated the type because the mode of action of this immunity system is different. There is one that cut, there is one that only do like RNA silencing, there is only one that do the activation of another gene, etc. So the goal of the CRISPR and Cas9 technology is actually to have this protein Cas as a scissor. You have this CRISPR sequence to recognize your DNA or RNA target, and then you cut it off. The mode of action known in CRISPR and Cas, if we can summarize, we have these two kind of option. We have MOA mode of actions option 1 on the left and MOA option 2 on the right. You have what we call the non-homologous end joining, meaning that you have this CRISPR and Cas9 construct and system going to your target cell and target DNA, and they will cut it into a double strand break. Why we call it double strand break? Because they cut the DNA double strand, so they call it double strand break, and the cell itself or the nucleus will try to repair itself. Because we have this proofreading mechanism, not like viruses that don't have, so they have many mutations, but in human, in plants or higher organisms, they have this kind of repair system. So when they repair it with non-homologous end joining, they have mistakes on the putting back the bases, the nucleotide bases. So these mistakes can delete of genes, can change the genes, and also can knock out the expression of a gene. So this one is very random, so that we call it NHEG. The other option that we have the same repair, but we use a directed what we call a template, a donor template. So we put another sequence among with the CRISPR and Cas9 system, and they will change with the template, so we can direct the change in the genome. That's why this technology called this repair is called the homology directed repair, HDR. And this second option is actually what we call today as the precise genome editing, because it's more precise and we change the base according to what we want. But CRISPR and Cas9 has known limitations. Of course, if you already work with this or you read about, you read in the papers about this, you know about the off-target effects, meaning that you have this target gene in your genome of your organism that have been targeted, can be cut in the right target, what we call it on target, but also in the other part of the genome that we doesn't want. Why? Because this technology has a very limitations also. We play with only small changes in the nucleotide bases. So we also increase this potential of target because they can recognize one or two bases change. I can explain to you in a few slides about this, but this off target has been a problem with CRISPR and Cas9, especially in plants. Why? Because in plants, in the genome, you have many duplications. You have allogam species. You have many similar gene family that can making this off-target becomes greater. You have also the problem of knockouts. And so the goal is to knock out the gene, make the gene not working again. But the problem is that in plant especially, when you have many duplicated genes, you have what we call compensatory expression changes of what we call redundancy. For example, you work with the Wilkie family, or you work with the NAC family, you have many hundreds of genes that are similar, have many domains of proteins. You cut one of them, the others can replace the gene that you have been knocked out. So the phenotype may be not seen, or you cannot see the phenotype changes. And in fact, due to the genome and environment interactions may be also not detectable. For example, here in the picture, you have to edit it. You have on the left is the wild type, and on the right you have the plants that already edited. You cannot see the changes because the goal is to change the genes that are responsible to the color of the leaves. The color should change into albino, right, or red or yellow, but not green. But here you can you can see that it's still green. Why? Because there is compensatory expression changes by the other genes. So this is the difficulties in CRISPR and Cas technology. And also in plants, but in human, the problem already passed with these difficulty deliveries of the transfect cell types. In plants, we have limited way to insert the CRISPR and Cas plasmid into the plant cell. In human, is very easy, but this kind of the problem is are known in the different organism that has been done using CRISPR and Cas. So we can talk about that in the discussion. In the discussion, if you like, I can explain more about the limitation of CRISPR and Cas9. So how about Indonesia? So honestly, if I have to say a few words about this research in Indonesia, well, I can say that we have these walls of CRISPR, or these walls of science in general. We have this complicated problem here in Indonesia in many aspects that can be solved using CRISPR, but eventually the research also expensive, because if the UK research scientists say this is not so expensive because they are talking about the range of funding, etc. But in Indonesia, it's very difficult to find funding. We have to go abroad, or we have to make a collaborations. True, it's expensive. I think you all agree about this. And it's a long way end. Long way end meaning that we have so many problems of outside the research aspect, and also in the research aspect itself, we have to be diligent. We have to be we have to go to the laboratory every day to see the monitor and to study. So it's very complicated. So that's why in this talk I would like to talk to you about how we break this wall. And our research has been funded since 2015 by the BPDPKS here, the funding agency for oil palm industry in Indonesia. We were collaborating with also Sokvin Indonesia in Medan, and PPKS, the Research Institute for Oil Palm in Medan, and also ourselves in Bogor. So we are trying to solve this problem because you live in Riau and Pekanbaru. Maybe most of you already know Riau is known as the oil palm plantations province. Many problems, but one of the problem is the disease that we call Ganoderma disease, basal stem rot. It's the rotting of the stem, sorry, caused by the Ganoderma boninense, like here in the picture. It's causing many problems in the oil palm, loss of productivity, loss of planting area, and also many other effects that caused by this small fungi. So maybe I can show you a little bit about how we can approach this research of CRISPR and Cas. The one that I told you about, the Ganoderma, is the one that we call it CRISPR-Cas research 4, or as a gene editing tool. We will need the genome information, we will need the candidate target genes, we will need the construct, how to design a single guide RNA or the CRISPR itself to go to the target organism and then cut the targeted genes. we have to find out how to deliver the system into the plants or into the targeted organisms, either human, either also animal. And also we have to be able to validate it through PCR, through NGS, for example. But now we also use this technology as a CRISPR-Cas research as a gene detecting tool used for COVID-19, for example, now, but developed before for Zika virus since 2009, 2012 by the same team from Fengzhang. We will need the genome information of the virus, for example, selected viral target genes, how to design the sgRNA and construct CRISPR-Cas, and they put it in one pot, what do you call it, one tube, and they call it pre-built tools. And you will have to amplify the DNA or RNA of the virus using a non-PCR system like LAMP, loop-mediated isothermal amplifications, or recombinase polymerase amplification, RPA. Then you join them together and you visualize using a lateral flow strip, and you will have this kind of detections, same as the rapid test for COVID-19. You will know that if you have a positive or negative infection of SARS-CoV-2. We can talk also about this in the discussion, but what I'm focusing now is the experience that we have in the laboratory about how we approach the problem of Ganoderma. So the goal is to find one susceptible, or not one, maybe multiple susceptible genes on oil palm DNA that are responsible to the tolerance against Ganoderma. And then we will try to find this gene and we will cut them off so that they cannot be expressed. And then we are expecting to have this DNA of the oil palm without the susceptible genes, thus leading us to the tolerance to Ganoderma. When you work on the CRISPR and Cas, this is what our experience told us. So you must have the possibility to handle both 50% of dry lab activities, 50% of wet lab activities. Dry lab activities, you must have an expert on bioinformatics, on genomics, on transcriptomics to work on this. And you must have teams also that can work on the wet lab, how to pipette, make clonings, put from agrobacterium, the system of CRISPR and Cas, into the callus or the targeted or the protoplasm of the targeted cells. You must be able to balance about these two types of laboratory works. And when we talk about bioinformatics, it's very complex. Actually, I showed you here is our research strategy that we have done to this research for the last 5 years. At the beginning, we don't have this genome information because, you know, in oil palm, everything is about to be, there was not much people that published publicly the genome of oil palm. So we have to find another way. If you don't have these genome informations of the target organism, you have to create themselves. For example, we did RNA sequencing, and also we validated using real-time quantitative PCR. And to get the candidate target genes. If you have the genome information, you can go directly, and then you have to design sgRNA using bioinformatics. And you have to construct the CRISPR and Cas9 using also bioinformatics. And you have to express the system and the delivery. And then you have to validate it using bioinformatics also. And then you have this field trial. Sorry. So this is an ongoing and very long way to do a research about the CRISPR and Cas9 using bioinformatics, but I will show you some of the examples. So these are the development of the transcriptome databases that we did. We collect many samples of susceptible and also tolerant oil palms, roots and leaves, many age of the plant, age years old, and the juvenile one, the immature one, 6-12 months old. We use a high-sec 2500 Illumina per N, etc. You must have the scale of this way to post-RNA analysis, like Tuxedo software suite for reference-based assembly. And then from this transcriptome database, you have to build, or you have to be able to take out the information from the DEG cluster. For example, we have more than 5,452 genes expressed at the same time during the attack of Ganoderma. So we have to be able to select this, because you only have a chance to edit 1 or 2 genes, or multiple, maximum 10 genes in one time of a plasmid of a CRISPR and Cas. So you must be able to select from these 5,000s, and then you make an Excel tables, very much, a lot of sheet there. You must be able to select one of them. So we select, for example, number 23 that are highly expressed in Ganoderma affected trees, because we believe that the genes that highly expressed in the susceptible trees are the gene that's responsible to the tolerance against Ganoderma. So we choose 3 of them, for example, here in the blue. And then we validate them through the real-time qPCR. So you take out the sequence, you design a primer for this, you do a real-time qPCR, and then you check if your hypothesis is right. For example, this one is highly expressed in the in the root system. So here we have this, briefly, we have these in susceptible plants. You have the highly expressed genes in the symptomatic susceptibility. So the plants showing symptoms, it's 6. And we check the expression of the gene, and we know that when we validate that what we found before in RNA sequencing has been validated in real-time qPCR. So we select this gene, what we call EGEMLP1, as the target of our CRISPR and Cas9 system. After that, you take out the sequence and you design a cRNA using one of the tools that exist in the internet. For example, Cas-OT, Cas-Finder. You just put the sequence there, put the reference genome, and then you select the target, and then it will give you a proposition of some of the sgRNA. And then you can order, or you can synthetically develop yourself the sgRNA. After that, you will need the bioinformatics to build the backbone of the CRISPR-Cas construct. You can use software Genius or SnapGene to just make the map of your construct. You put your sgRNA sequence and you try to develop yourself. If your laboratory has the ability to develop this or to do synthetically, you can do it. You can just synthesize it. But if not, you can order from the other company that they have open also for this kind of work. And after you have this CRISPR and Cas9 plasmid, you have to put it into the organism, the target organism. You must have good transformation system. We check first our transformation system using agrobacterium, because why in oil palm we cannot have this protoplast transformation system. It did not work yet, even in the world. So we have to use this agrobacterium system. We check and we validate the efficiency about 20 and 60%. So now the model plant, we use the gene from sugarcane just to check. We put it in the sgRNA and we try to cut it. And now we have this plant edited of P5GS genes responsible to drought in sugarcane, and the transformation system works very well. After that, we try to check this, use the transformation system on the oil palm cali. So we have to select hundreds of thousands of callus of the somatic embryogenesis. And we have to select the one that has the good color here, friable callus. We have experts to do this. So they are selecting the callus, doing what they know and their expertise. For example, we select this one and this one and this one, and we just cut them, put them in another media, and we try to grow them in the media using hygromycin. So the callus that grows in the hygromycin is the one that survive and also has the CRISPR and Cas9 plasmid in their genome. And this one is not. This one is also a very laborious work and quite demanding of time. And after that, we haven't done this yet in our laboratory, but in the world, they suggested us to do what we call mitigation of sgRNA, because sgRNA has this tendency to do an off-target. So what you do is actually to sequence your callus. For example, if a plant cell, maybe your stem cell, you need to sequence it and you try to check in the area of the genes, can you find any off-target? For example, if I can show you here. So you have this gene of EMX1, this is the read, the 4572, this one is the one that correct, on target. You have no differences in the bases. So, but you have other, like 3000-ish reads, 1000-ish, and many others that also cut. So that's why this sequencing, or what we call mitigation of sgRNA, is very important. But nowadays we don't have this kind of system in Indonesia, for example, because it's very expensive. You have to sequence the callus only to check this, and after that, eventually you have to go forward to your experiments to grow the plant and just prove it in the field. So this is an option to do, but many scientists in the world suggested other scientists that work on CRISPR to do the sequencing of the edited callus or edited cells after they has been proven to be inserted by the CRISPR. And I would like to show you just a little bit how we detect the CRISPR and Cas9. We use detection using DNA and also RNA. Why? Because as you can see here in brief, the DNA of many lines of our callus did not show any changes, right? So using EMLP primers designed for DNA. But if you look in the RNA using a primer from the RNA, you will see that the lines number 4 and number 6, you don't have the expression, or even number 5, 7, and 8, you have a very low expression of the gene. But what we call KO is the line 4 and line 6. You have the DNA intact. They are changed, the bases change, but they create something inside the DNA that cannot express the protein. So we sequence this callus, and we see that when you see the edited calli here, we are creating the codon stop. I hope that it can be seen. There is a star here. After CCAA, GT, CT, AG. So in A here, you have a stop codon early in the beginning of exon 1, meaning that the protein cannot be developed. So that is why we cannot see the expression in the RNA. So the CRISPR and Cas9 system works very well and make the genes of what we call EMLP1 KO in the oil palm. But the next problem is to prove this theory in the field, because now we are planting the plantlet in the field on acclimatizations, and also we would like to find out in the next forthcoming years to validate this theory, because you can have this whole range of experiment in CRISPR, but finally you don't have a good result in the end. So it could be a pity, but we are trying our best. So the facts and complexity of CRISPR, as you can see when I explained to you, is quite complex. I mean, in plant, for example, you need NGS analysis to do or to find out the genes that you are targeting. You have many, you have thousands of genes, but you need this kind of NGS analysis just to show them 1 or 2 genes that you are willing to target. And then in plants, especially, you have lower transformation efficiencies, not like in humans and also in animal cells, quite be easy. And in theory, you will have this permanent mutation, but you need research. Nobody doing this research for 10 years, 15 years, just to check if the mutations happen in the plants inherited for the many generations to come. So this one is actually the complexities. But the facts about CRISPR, yes, it is better than the previous technology of recombinations. We have to work with this complex genome structure in plants, but in human, it's more of clear ethical boundaries, because we don't want to create babies, for example, you know, the problems of a human baby edited by CRISPR. We are afraid if we are not doing right, we can create what we call a baby Deadpool, maybe. So very dangerous and very unethical. So, but in general, genome editing using CRISPR and Cas9 is still less cost and less risk than the conventional transgenesis. to end my presentation, really sorry to take about your time. In the upper level of the tip of the CRISPR, what we call, we know that CRISPR can be used as to edit babies, to knock out genes. Now we find anti-malaria also for detection tools in COVID-19, in plants to knock out genes. We have these different type of CRISPR and Cas9, 12A, 13A, DCAS, etc. We are very excited. But beneath what we don't know yet is that if this technology has an unimaginable benefit, like end of the disease, but creating another horrifying tools for edit something that we don't have, that we don't know yet until now. So if I allow to quote the ethicist Paul Afterbaum, that as we begin to able to edit the gene pool, it may be the case that we can anticipate some of the consequences, the changes that we are making, not now, but maybe in the next future or generations to come. So I would like to thank my team in our laboratory. We have so many students also, so many research scientists and good collaborative work. And I am open for questions and also for discussions. And thank you for your attentions. And I return my time to Ms. Rani. Thank you.
Thank you, Dr. Riza, for a very interesting talk. Dear all participants, we have learned about what actually CRISPR is, its mechanism, its validation through bioinformatics, and how it's applied to his research, and also about edited babies. Kepada seluruh peserta diinformasikan bahwa jika ingin memberikan pertanyaan nanti pada sesi tanya jawab, boleh klik raise hand fitur, kemudian nanti akan dipanggil untuk memberikan pertanyaan secara langsung. Boleh dalam bahasa Indonesia ataupun bahasa Inggris. Bagi yang ingin menyampaikan pertanyaan lewat chat juga dapat disampaikan lewat chat. Untuk kehadiran, mohon diisi linknya, dapat diambil linknya di kolom chat. Dr. Emma, are you there? We're going to check your connection and speaker. Hello. Okay. We're gonna move to, I would like to welcome Dr. Hanan Hassan as our next speaker. Dr. Hanan will talk about metabolic engineering and CRISPR-Cas system in industrial biotechnology. He is a lecturer at Faculty of Food Science and Technology, University Putra Malaysia. He is also involved in Halal Product Research Institute, European Biotechnology Network, Malaysian Society for Microbiology. He holds his PhD on metabolic engineering from University of Sydney, Australia. Now he is a leader of a project on metabolic engineering and CRISPR-mediated genome editing to increase the yield of microbial valuable compound. Dr. Hanan, time is yours.
Rani, can you hear me?
Yeah,
Can you
yeah.
hear me?
Yes, ma'am.
It's okay. Let's talk.
Thank you, Prof. Emma.
Should Prof. Emma go first or I go first?
You go first. It's okay.
All right. Thanks, Prof. Emma.
Yes. You go
Okay.
first. Okay.
All right. Assalamualaikum and good morning to everyone. Okay, let me share my screen first. Okay, first of all, thank you, Ms. Yuli Haryani, for the introductions. I would like to express my gratitude to PBBMI, the Indonesian Society for Biochemistry and Molecular Biology, I would like to also thank you, Dr. Ramawati, for the invitations and having me for the research talk for today. Okay, I'll just go straight away to my topic for today. So today I'm going to be talk a bit more casually about my research, okay, which is mostly my research evolved around metabolic engineering. So today I'm going to share with you guys and discuss as well with you guys the application of metabolic engineering and the use tools of CRISPR-Cas9, which has been discussed in detail by Dr. Riza, for the production of valuable product in industrial biotechnology. First of all, let me just quickly introduce myself. Rani has introduced me. My name is Hanan Hassan. I'm currently serving as a senior lecturer in University Putra, Malaysia. Okay, all right. Okay, so what I want to discuss today, we're gonna highlight a little bit about what is exactly biotechnology. I'm sure everyone here has a background of biology or biotechnology, how the emergence of biotechnology revolutionized our industries, okay, be it for pharmaceutical, industrial, and food and agricultures. And then I'm gonna discuss about genetic engineering, how actually genetic engineering has impact us, and honestly how it make our human life and the world so a much better place. I'm going to compare that with the emergence of new, I would say it's new, but the latest technologies, which is metabolic engineering. And then I'm going to discuss briefly what is exactly CRISPR. Probably I'd have to discuss in detail, since the results I've discussed in detail. I'll just mention about it briefly and simple mechanism, and the application and potential application of CRISPR-Cas9 on the production of valuable products. So exactly what is biotechnology? Biotechnology is actually a key to the bioeconomy. So discovery of biotechnology is the enhance of human life. It is actually, introduction of biotech is actually to make human life much more easier and to improve ourselves, and not just ourselves, animals, our food and crops, our environments, the world as a whole. So the application of biotechnologies is able to produce proteins for therapeutic and vaccines. As you all know, and Dr. Riza has mentioned as well, the use of CRISPR-Cas9 to combat COVID-19. So due to the idea and tools that is available in biotechnology, we're able to combat this COVID-19, and we are coming so much closer in having a vaccine to combat COVID-19, hopefully, right? So we have also DNA fingerprinting. DNA fingerprinting is actually very important for us to know our hereditary, our phylogeny, okay, where organism comes from and all that. It also revolves around research on genome structure and function for us to understand how a certain gene works. Modification on food and crops, this is improvement on food technology and crop technologies. Okay, for this one, when it comes to biotechnology, we not just simply, when we say genetic modified organism or genetic modified food, it's not about only seeing one tomato, red colored tomato, or you have a big size, pumpkin sized tomato. It just not revolve around that. We talk about the idea of having to have much more nutrient tomato. We talk about on having to provide more nutrient, especially in third world country in Africa. So the emergence of biotechnology help us to achieve that. Products of biotechnology, we have organisms with desired biochemical morphology. This means that we're able to improve a certain strain to have a certain characteristic that's beneficial for it. This is, for example, in terms of fermented food. You have some microorganism to produce fermented food that is much more nutrient, longer shelf life, and very high characteristics. Okay, organisms that are able to produce valuable products. This is what I want to highlight today. Okay, a lot of products that have been produced nowadays is through chemical synthesis. As we know, yes, chemical synthesis can be very efficient. Sometimes it can be cheaper. However, it's not sustainable. We've seen a lot of pollution nowadays being made through chemical synthesis, which we want to replace this to biotechnology using microbes. And as well, this is some of the information about tissue or organism or tissues that are unknown. Usually when we study this, we highlight about the rare disease that occurred nowadays. For example, Alzheimer and Parkinson, we still study about that. We still haven't discovered any particular therapy or drugs that are able to cure these. Okay? This is one of the things that biotechnology is able to address. So let me just highlight quickly what is exactly genetic engineering. Okay, genetic engineering is the process of using recombinant DNA. This is the tools under the field of genetic engineering to alter genetic makeup of an organism. For example, we want this organism to have longer life. We want this organism to have less toxicity. We alter that certain genetics to obtain desired phenotype. Phenotype means a physical appearance, or want to this particular microbes or certain cell to have particular physical design. For example, if we were to apply this into humans, probably going to have humans that is much taller, more resistance to diseases. We're able to combat any type of flu. These are the things that we desire. Example of tools that involve, this is what we call classicals. We have random mutation, precision enzyme, plasmid insertions, homologous recombination. Although all this still being used nowadays, genetic engineering in terms of these tools are what we call is a old school or last year news. It's come to a certain extinction. That's the reason why we're moving to new technologies. So this is what we call metabolic engineering, the comparison of metabolic engineering and genetic engineering. Okay, let me just define what it meant by metabolic engineering. Metabolic engineering is defined as a purposeful modification, meaning that you did that modification on purpose on metabolic pathways. So we have a network of pathways, which I'll show after this, to obtain efficient carbon flow for the production of industrial valuable compound. This is stated by Gregory Stephanopoulos at MIT. He's actually I would say, from my background, the father of or pioneer of metabolic engineering, which actually been discovered not in the year of 2000, somewhere like that. Okay, so the idea of this, if I want to do a layman's term or metaphorically, imagine what we have on the right hand side. This one, your right hand side, is a complex metabolic pathways. You will see this is like a traffic highways or a metro complex or your subways, which is interconnected. Yes, it's interconnected. You can get from point A to point Z, but within that point, you have a lot of diversions. Okay, this is what diversion of carbon flow, which we can see on the left hand side, is like a car which represent your carbon flow. You want to get point A to point B, sometimes you have to reroute yourself. Okay, if it's traffic jam, if it's too congested, meaning that your travel from point A to point Z is not efficient, right? So what we want to do in terms of metabolic engineering, we want to turn this complex pathway to this one. All right, we want to have an efficient carbon traffic flow. On the left-hand side is actually called Autobahn. It's actually highway in Germany, which is very efficient, very safe, and a lot of cars travels here can reach to about more than 200 kilometers per hour, which is very, very efficient. So what we want on your right-hand side. So every time we feed a nutrients in here, we want the nutrients for transfer from point A to point Z efficiently, without any diversion, without any influence or other metabolic, or we call that byproducts or other pathway that we need to divert to have a certain products. So the idea is we want to shut down all these called it noises pathways so that we can get from direct point A to point Z quickly, more efficiently. That will give us a lot more profits and efficiency. So that's the whole idea of metabolic engineering. What I want to highlight as well in terms of genetic and the comparison between genetic engineering and metabolic engineering is the idea of genetic engineering. You only modify one or two genes at that particular site without looking at others. Where in a real case, in reality, whereby when it comes to microbes or any kind of cell, everything is interconnected pathways. Meaning that if you knock down or you overexpress a certain gene or you modify a certain gene, it will affect other genes as well because everything is interconnected. So this is metabolic engineering. The idea of metabolic engineering is the improvement of cellular activities by manipulating enzymatic regulatory transport function and the cell with the use of recombinant DNA technology. This is the tools of it. It's actually multidiscipline, focused on metabolic pathways, it regards to combination of omics studies, transcriptomics, genomics, metabolomics, proteomics, and all that. It's efficient approach for strain information and process optimization. I'll explain in detail after this. It has been successfully applied in micro production of various chemicals. A lot of journals have been nowadays have been published in terms of metabolic engineering. Metabolic engineering as well have its own journals, which I think is one of the highest journal in biotechnologies, which has impact factor close to 9. Okay, so it provides strategies for systematic metabolic, could be categorized into two groups: either rational intuitive, meaning that you study about that, you analyze about the patterns and everything, or you have systematic and rational random approach, which by through to approach of trial and error. Both approach are quite efficient in terms of metabolic engineering. Okay, let me share you this diagram of the idea of how metabolic engineering works. Sorry, apologize. Okay, this one. Okay, so when you are trying to, this is an example of production of biofuel from carbohydrate, be it in terms of corn or from biomass. Biomass is actually a waste. Okay, how do you turn waste, the idea of methodology, how do you turn waste into efficient, valuable products? All right, first on this side, design. We need to understand, before we able to convert waste into a valuable product, we need to understand the host metabolic pathways. Is it able to produce biofuel on its own? If yes, how much it produce? The idea is if it's able to produce, how much will it increase? So that entire thing leads to design. We need to design the pathways, metabolic pathways, understanding its whole metabolic pathways, what substrate does it use, and what sort of fermentation cultivation does it covers. Okay, in terms of cultivation parameters, this also involve fermentation by processing. You have to control its pH, type of substrates, temperatures, agitation, and all that. That relates to what are called parameters. So once you understand the whole metabolic pathway, you want to go from point A to point D efficiently. But on your way, you have to have, on your way, there's a noise, we call noise pathways, which is D. We don't want D. This is useless. So what we should do, we eliminate the D. Once we understand the whole pathways, we move on to building it. We're building it using the tools. One of the tools that I'm going to highlight after this, which is CRISPR-Cas9. Okay, one of it. Okay, so cloning technique, engineering of hosts, these all the ideas and theories they're able to improve. It is called theories, and testing the theories, we're able to improve the strain to produce biofuel more efficiently. Okay, I'm going to discuss after what I mean by efficiently. Okay, and then once we're able to build this host, then we test it. We put into a shake flask, we test it. How much it increase? If it doesn't increase, how much it reduce? If it increase, if it reduce, maybe something wrong with the build. So we go back with the build and go back to the testing. If it increase, now we go to analyze. We understand, okay, how much increase? What's the rates of expression of certain proteins? If it doesn't increase, what's wrong? Sometimes you might the metabolic the gene itself, you increase to a certain point, but there are other limiting factors that might limit the production or increment of your desired products. Okay, so once we analyze this, is it good enough? Does it increase? If it increased by 10%, is it called good enough? If it's not, if you want from 10% to 100% or 1000% improvement, we have to go back to the design. So it's actually a rational approach through trial and error. So it's like a circular motion. Okay, the idea of it, at the end of the day, we want to improve our products as much as we can from, if possibly, use a carbohydrate from a waste, a biomass, a molasses, whatever that you want to call that. And then once we achieve the possible highest product or yield that we can get, we put into a bioreactor, a bigger scale of bioreactor, from 100 liter to 1000 liter to 10,000 liter working fuel. Okay, when I talk about the efficiency, you imagine this. If you were to given a certain workers, if you were given to a certain type of workers, you spend about 1,000 ringgit. You expect them to produce 1,000 type of products, right? 1 ringgit per product. However, they are within the type of workers, someone that might not be efficient. Okay, someone that might produce half a product, someone that might produce less. So reality, we spend 1,000 ringgit, but we get like 50 products. Okay, the idea of metabolic engineering, we want to spend as little as we can for that 1,000 ringgit. Possibly, you want to get at least 500 products. If possible, we want to get into 1,000 products. We call it efficiency. In terms of microbes, every time we supply them with 10 grams of biomass or carbohydrate, we expect to get as close as to 10 grams per liter to amount of product of biofuel. This is called efficiency, increasing the efficiency. Okay, this is the type of engineering metabolic pathways. As you can see here, every time we feed a glucose, they have to go through a lot of pathways in between to get into your desired product. The idea of this, we want to get from point A directly to a certain point, for example, to acetate efficiently, without having to go through other type of pathways. There's an example whereby some of these red represent knockout genes, and green is an example or representation of overexpression. So all this knockout means that you're shutting down a certain pathway, and then you increase a certain pathways so that you get from point A glucose, which is a substrate, to point B to point Z acetate more efficiently. Okay, these are called breakthrough in genome editing. Yes, actually, genome editing has been discovered long time ago, even 1989, but it's being well documented in 1989 by homologous recombination mediated targeting. Don't be surprised, homologous recombination is still and will always still being used up until today. And then move on to the year 1992, 1998, and 2000. Okay, before that, we have a Krylog system. Krylog system is similar to Cas9, whereby it has a protein that able to nick a certain type of a certain area of genetics or genomes to modify that particular places. Okay, and then in year 2000, it's been this bacterial CRISPR-Cas9 has been discovered, and then at year 2009, we have transcription-like effector nucleases called talents. Discovered in Xanthomonas, and finally in 2013, CRISPR-Cas9 is discovered by Jennifer Doudna and her team. Okay, so this is what we call a timeline where how CRISPR-Cas9 is the latest technologies for genome editing. So why exactly that CRISPR-Cas9 is famous, like Dr. Riza has mentioned nowadays? From 2013 to 2020, the booming of the publication of CRISPR-Cas9 is exponential. A lot of people have been focusing on CRISPR-Cas9 because it is efficient. The understanding of CRISPR-Cas9 is it able to edit genomes or sequences up to one base pair. That is very efficient. It's unlike we have seen before. It is basically programmable nucleic acid targeting tools, which Dr. Riza explained it before. You can target is very precise. Okay, it is potential for the many microbial engineering applications, not just microbial, from cell line to crops to human cell lines. Okay, it's precise editing. You can do upregulation or called overexpression. You can downregulate, you can knock out, and you can insert. I'll explain after this in terms of mechanism. It can able, of course, due to this and what we want to achieve in metabolic engineering as well, significantly increase yield of desired products. Okay, apologies for the misspelling. And it is what it is. It's very simple. It only use a Cas9 and D-Cas9 system with a single guided RNA. With these two, it able to target any type of gene as many as one base pair. Okay, this is the mechanisms. I'll go into a bit differently. This is sourced by New England Biolabs, NEB. So in terms of this, you can do insertions. Okay, the idea of genome editing using Cas9, where Cas9 able to detect any PEM sites. Okay, these PEM sites, once able to detect that site, it will dock into a genome, and within that, it will upstream or downstream of these PEM sites, it able to cleave that particular targeting sites. And with that, once we have this, we can do whatever we want to here in between in this space. Either we insert or delete by inserting antibiotic markers, Or what we can do, we can insert a new donor DNA. For example, what we delete is an old promoter, and now we insert a new, much stronger promoter. And as well, what we can do, we can cleave or nick into this target A genes. Once we, for example, this gene, particular genes is what we call an unwanted genes. What we want to insert a new products. Okay, for example, what have been discovered nowadays is the production of biofuel through algae. What they have done, they're cleaving a certain type of genomes and they insert a new genome to increase the efficiency of production of biofuels. Okay, and lastly, of course, localization with defective Cas9 or dCas9, where you can have activate, meaning you increase or turn on or increase at the same time its regulation of certain genomes. You repress, meaning that at the same time, you want that particular gene is important or essential for the growth of microbes, but you cannot delete it. So what you do, you repress it to a certain point where it doesn't kill the DNA, it doesn't kill the host, but at the same time, it gives much more efficient metabolic pathways. At the same time, lastly, you have visualization. This is being used for genome study, where you want to know at what particular site of that particular DNA or genes you want to study. Okay, this application of CRISPR, very simple figures I want to highlight. It can be used in cells, animal models to improve. Yes, nowadays we are research more into improving our animal life, especially those that involved in agriculture, poultries, cows and buffaloes. You want to improve their disease. You want to improve their resistance to a certain disease. Okay, of course, human disease therapy. Example, we want to combat rare disease cases, Alzheimer and Parkinson. Of course, nowadays, later on, you want to develop a vaccine using CRISPR-Cas9. New drug screens. Okay, you're able to produce any type of drugs. For example, now that are usually nowadays we still produce some of the drugs to chemical synthesis. And then we have unscripted and epigenetic edits. This is where we want to study a certain genome of a certain microbes or cell lines of humans. We have agriculture and fruit. The improvement, this is what I want to highlight, the improvement of food safety and quality. We want to improve a certain characteristics of nutrient of certain food, especially in fermented food. The idea we want to produce a fermented food that is much more nutritious. Embryo and fetus gene editing. This is to avoid any genetic disorders prior to giving birth. So you can say goodbye to cleft lip. You can say goodbye to Down syndrome and anything that is genetic disorders. Of course, nucleic image and detection and diagnosis. This is very useful when you are studying about the function of genes. Okay, this is what it calls a strategist, a very simple one. Strategist design genetic manipulation. You want to, first you have to know which sort of genes that you want to highlight or you want to target, you want to knock out, or you want to repress, or you want to upregulate, okay? Once you have that, you have to select your expression systems, okay? The thing that we use in our lab, we use dCas9 with single-guided RNA in each respective plasmids. And then you have to select your target sequence and design your guided RNA. You have to know your cell line organism, different cell line, different microbes, and different crops or in agriculture. They have different systems, so it's not tailored to each. It has to be tailored to each type of microorganism or organism. You have to select a gene and a genetic element to be manipulated. You have to understand the whole metabolic pathways. You have to know your single-guided RNA based on predicted on-target and off-target activity. The software that we use is through BenchLink. Okay, synthesize and clone desired guided RNAs. This is what usually we use this onto a plasmid, which I'll explain after this. And we deliver the Cas9 and guided RNA within the host. And then once we deliver this, we know that we can target either knockout or upregulate certain genes. And finally, we validate genetic modification to phenotypically or genotypically. Okay, these are some examples of application of CRISPR-Cas9 that has been used through CRISPR-Cas in metabolic engineering that has been studied and done before. Okay, in terms of I've separated into three part of fields. In industries where the production of biofuels. Okay, some of the biofuel, like I said, they are testing it in, they are experimenting in algae for the production of biofuel. I think Exxon and Mobil have been aggressive with these studies. I think they are coming very, very close. They actually have successfully able to produce biofuel from algae. But if I'm not mistaken, the yield that they produce cannot compete, as for now, cannot compete with the petroleum, the natural petroleum. You have phytochemicals, okay? All these phytochemicals that are very, very useful in the production for industry. You have medicines, you have activation of tumor suppressor gene. This is for cancer study, by Torres Ruiz. You have protecting CD4 cells for HIV, it is for HIV infection. And one of the studies that I've did for my PhD is I able to increase levastatin yield in Aspergillus stress. Levastatin is actually one of the highest use of, or statin product is one of the highest used product in Malaysia, matter of fact, in the world as well. It's to combat cholesterol. It's to use to reduce cholesterol level. If I'm mistaken, the revenue by this one company, I think Merck company, the revenue per year in the US alone is about $4 billion. Okay, and food agriculture able to improve yields and quality of our crops, develop abiotic stress, and able to develop a tolerant crop much more to resistant to pesticide and insecticide. And finally, our ongoing studies, which we want to produce, we want to increase the production of bacteriocins, which is particularly niacins, in the host of Lactococcus lactis to be used as biopreservative. As for you, if for you which not familiar with bacteriocin is what we call a biopreservative. It's similar to chemical preservatives to extend shelf life of a food. Okay, this was a simple metabolic pathway that our team have designed. So the idea of this project is we want to increase the precursors. Okay, one of metabolic engineering strategies is to increase the precursors of niacin for us to increase the niacin yield at much higher amount. So the precursors of niacins, okay, which is cysteine, serine, and threonine, all these are amino acids, okay, because bacteriocin or niacin is actually a peptide, a biopeptide. So it requires a certain folding of these three compounds to produce a called methionine. Okay, so the idea of it, we want to increase this by highlighting a certain gene. If you see certain gene here, okay, these are the things that blue here, color here. These are the genes that we want to highlight or increase its expressions. Okay, those that we target X here is all the things that the genes that we are trying to knock out, so that the transfer of glucose to produce cysteine, serine, and threonine are efficient without ever to go any other pathways. Okay, at the end of the day, once we have these, we're going to focus on the maturation and the production of niacin. So at the end of the day, we're going to have higher productions or higher yield of niacin. So these are briefly our strategy. So we have designed, our team have currently designed two types of plasmid. We have Cas9 and Cas9 plasmid, where we insert into a certain plasmids. And then the other plasmid, we use two plasmid, and then the other one we have single-guided RNA plasmid, where we insert our target of genomes for knockout and for upregulation, and we transform it into our host, which Lactococcus lactis is ATCC products. This host is actually able to produce niacin, but not in a very not in a very low amount of yield of niacins. Okay. As for now, the reason why niacin actually is accepted by FDA to be used in food products. Okay. So the idea of it, we want to increase niacin at much higher yield by targeting a certain gene that I have highlighted before. So in conclusions, okay, in terms of CRISPR-Cas9, CRISPR-Cas9 is actually one of the latest tools that we want to use, that I'm sure everyone to use in my background expertise in metabolic engineering to efficiently target a certain gene to get our desired products. Okay, in terms of this, in terms of application of this one is endless. We can use from bacteria, yeast, fungi, and algae, not to mention we can use on crops, in cell line, and of course in humans, for us to have a better life, for us to have a better sustainable environment, and for us to moving forward and having much more safer food and more quality foods. Okay, with that, thank you very much.
Thank you.
Okay, this is a poster. Maybe we can talk about this later.
Thank you, Dr. Hanan, for the interesting presentation on the history and application of genome editing and metabolic engineering, and application of genetic engineering, as well as the CRISPR construct for any variable compounds. Now we continue for the question and answer session. Mr. Rahmat, would you please help me to see anyone raising their hand to ask some questions?
It might have skipped me, but not at the moment.
All right, from the chat column, Prof. Titania sent us the first question to Dr. Riza. Prof. Titania, are you going to ask directly, or you want me to read the question?
Okay, I can ask directly.
Thank you.
Hello, Dr. Risa Putranto. Thank you very much for a very good lecture. My question is, what if the palm susceptible gene to Ganoderma that you are knocking out, or that you are cutting out, is important to the productivity of the oil bunch, or maybe have important other function? Do you check on that too, or maybe you can elaborate more on this problem?
Yeah. Okay. Thank you, Bu Titania, for the questions. It is an interesting questions. Everyone asks, also ourselves in our laboratory. For now, we have targeted 5 genes, 5 genes in oil palm, especially involved in the plant defense mechanism. We see many, so that is why we were doing RNA sequencing, and we combine also with the genome sequencing from the publicly accessible genome of oil palm, and we check if there is any relation between the plant defense mechanism that we are targeted with the productivity of oil palm. Until now, we are not seeing if this one of our many genes of plant defense mechanism has the relations with the productivity, because we are afraid. I understand the question, because we are afraid we are trying to knock out one genes, for example EMLP1, and then we will have this oil palm tree have a good tolerance against Ganoderma, but do not produce any fruit bunches. So that is one of the concern. But until now, we know that by study in silico and also by study of gene expression, they don't have any relation, meaning that negative relations, meaning that if you knock out the target genes, you don't have yet, we don't have yet the information about the productivity, because for right now the plant is in the nursery, and next year we would like to try to perform what we call field test by injecting Ganoderma into the soil and also etc. to find out if the plants really have these traits of Ganoderma tolerance. I hope I answered your questions. Thank you.
Yes. So still a long way to go.
Still a long way.
Good luck. Yes.
Yeah. Thank you. Thank you.
Thank you, Prof. Titania and Dr. Riza. The next question is from Rudy. Rudy, are you there?
Yes.
Okay, thank you.
Okay. Thank you, Rani. Thank you for Dr. Hanan for the good presentation. I have a question for you. Um, in terms of the role of CRISPR, is there any roles in drug discovery? If there any, can you give us some example? I think I read in one of your publication regarding to, is it simvastatin or something like that? I can't remember. So I mean, based on your expertise or your experience, there any roles in the roles of CRISPR in drug discovery? I mean, thank you. Hello.
Thanks, Rudy. Dr. Hanan.
Is it addressed to me or Dr. Riza?
It's you.
Oh, okay, it's me. Yes, Rudy, yeah. Yes, actually, a lot of studies have been done nowadays on the drug discoveries for CRISPR. It's not new. CRISPR is a tools to be used in for production of certain products. Because a lot of drugs, actually, if you understand about the discovery of drugs, most of it comes from natural products, right? One example is the oldest one is penicillin. It's come from penicillium. So a lot of product nowadays comes from natural product that we are trying to put all these genes within a certain host, usually microbes or any kind of fungal or yeast to produce these kind of drugs. So the use of CRISPR make this transfer of discovery of new drugs and transfer production of new drugs into a certain host much more efficiently. And as Dr. Riza have mentioned before, vaccines, especially the discovery of vaccine of COVID-19, yes, a lot of people and a lot of studies, publication nowadays, they have done in terms of how you are able to establish or create a vaccine through CRISPR-Cas9 as well. Dr. Riza may want to highlight that or may want to add that in more detail, I suppose.
Okay, so means that is more in terms of the production of the metabolites. Is there any like can we do modify the structure using the CRISPR?
Yes, of course. Within using CRISPR, you can study a certain function of certain genes. Before you want to have the discovery, you have to understand how it works. Okay, this all related to molecular work and molecular biology, whereby you have to understand how certain genes or how certain drugs able to elicit its function mechanisms. So before we can call it a drug, we have to understand how it works first.
Right.
Yeah.
Okay, thank you.
All right.
Thanks,
Maybe Dr.
Rudy.
Riza want to add, I suppose.
Yeah. Thank you, Dr. Hanan. Well, actually, in what has been explained by Dr. Hanan is correct, because as long as my experience working on CRISPR, we don't have yet truly that CRISPR can modify structures. But most of the studies were being done on how the genes working, how the mechanisms are known, etc. How if you edit one gene and then you have the possibility of other chemicals and other substances, that were the study. Because for now, even when you develop CRISPR and Cas9 for detection tools in COVID-19, it takes many years actually. The one that you've seen today in publications or in the media is the one that there has been research for it for at least the last 5 years, because the first publication about the COVID-19 or CRISPR detection tools was in 2015. So it was started there. Sorry, 2012 even, 7 years ago. So it is not a sudden. Why? Because CRISPR and Cas basically is the technology that you use to edit. So if you want to edit structure, because that was more in the next aspect, because you have a molecular biology dogma. For me, CRISPR and Cas9 lies on the aspect of DNA, RNA, and also mRNA, in betweens. But if you want to modify proteins, I don't think that you can use CRISPR and Cas technology. I hope I add the explanation from Dr. Hanan. Thank you.
Thank you.
Thank you, Rudy, Dr. Riza, and Dr. Hanan. I found here that Arfianti is raising her hand. Are you there going to deliver the question? Arfianti?
Do we still have time, Rani?
Okay.
Have time?
Yep, of course.
Yeah. Okay, thank you very much, Dr. Riza. I think I will repeat my question. So I'm Arfianti from Universitas Riau, Faculty of Medicine. So as you mentioned that the CRISPR, that CRISPR is very promising, but still yet it's very expensive, especially in the Indonesia setting. And also for the complexity of the research in human, we have lots of issue regarding the ethical issue. So how about the CRISPR technology in animal experimentation? Because I know in overseas, they already use this technique to produce the animal models rapidly, because usually if you use the conventional genetic technology, you need years to produce like a knockout mice. But with this CRISPR, you can shortcut, or you can get the animal, the knockout animal, quite fast. So how about it? Do you think that this is quite promising to develop in Indonesia? As you know, for drug discovery or drug development, you need to do the clinical studies. So I think this is very promising to develop in Indonesia. And for Dr. Hanan, can I ask two questions, Rani?
Okay.
Okay. So for Dr. Hanan, as you mentioned that the metabolic pathway, because I used to work with the insulin and mTOR, mammalian target of rapamycin signaling. And usually it is very common if you suppress one metabolic pathway, and then you can get another like compensation from other metabolic pathway. So can you give advice if we interested to like learn about one metabolic pathway, and then you want to do like separation to see what is the function of the metabolic pathway? What sort of like strategy that you need to apply before you can make sure that this is the correct way to do that on research, usually for the human studies, for example? Thank you very much, Rani.
Thank you. Dr. Afrianti, would you please, Dr. Riza, answer the first question first?
Okay. Thank you, Dr. Arfianti, for the questions. Very good questions. Maybe if I allow to share a slide, it could be better to explain, because I did this one also. So in human, it's actually going like a fast car when you try to do a CRISPR. When I said human, also the animals experimentation on CRISPR is also in a fast car. You are right. You said about the clinical, the test of preclinical studies using animals. They have been tested using as mice, knockout very fast. Zebrafish. They have rats also. They have mammalians also. Very quick. Normally they use as a therapeutics agent. What I'm talking is the CRISPR and Cas system. The problem is, in every discussion, I did this discussion with one of the biggest company in Indonesia. many years ago in 2016. We were all talking about this and the possibility of doing this in Indonesia. And the problem, we have this huge wall of bioethics. At that time, that was very interesting. But in 2017, if I'm not mistaken, Dr. He Jiankui presented his edited baby, and that destroy everything. Why? Because now people are aware about doing CRISPR and Cas9 experimentation in animals, especially in human lines. It's very dangerous, they say, because you can create one organism living, not having the receptors of HIV, but we never know if this baby grows up and then he or she would marry some other human being and they have child, and we don't know that. The fear in the society push back the CRISPR research in humans and in animals. But I think if in Indonesia myself, I haven't not yet seen any regulation according to CRISPR against animal. Human maybe not, because human maybe we follow the international regulation, but animal not yet. And I think, in my personal opinion, if we want to do research in animal experimentations in using CRISPR and Cas in Indonesia, it is very possible and very promising, especially like now we have this COVID-19 with the need of more experiment in preclinical using animals. So we can push this and we can use CRISPR-Cas to study the effect of genes, effect of a vaccine, for example. If we knock out the receptor of one gene in animals, can this vaccine give more benefit, or there would be a drawbacks of the vaccine, etc. But yes, I think it's very promising. The problem is you need you will need a stable funding for this, because CRISPR and Cas9 in Indonesia, for my own experience, is not cheap. It's quite expensive actually. So this should be the main concern if you want to you are willing to do this experimentation. But if you find something new and it's very promising, you will discover many new things using CRISPR, because the technology is not yet finished. It's just the beginning. Like I said, it's the tip of the iceberg, and below we don't know yet what in the future that we can do using this technology. I hope I answered the question, Dr. Arfianti. Thank you.
Thank you very much, Dr. Riza.
Okay, Dr. Riza. Dr. Hanan, would you please answer the second question from Dr. Arfianti?
Okay, thank you, Dr. Fianti, for the questions. Actually, a tough questions.
Tough questions, yeah.
In terms, yes, I'm just joking. Yes, of course. In terms of any production of therapeutic drugs or insulin, I'm not sure what sort of advice I can give it to you. But the idea of having to increase the idea of metabolic engineering is not only focused on increasing the yields. Sometimes other people have been working on the yield itself to a maximum point, meaning that that's the limit that the insulin can go. But in terms of that, you can still manipulate into the substrate that being used. As I understand nowadays, if I'm not mistaken, there's one study whereby they use a bacterial microbe. I'm not sure what bacteria, whereby they grow this microbe within a milk sac of a cow to produce insulin. So they engineer a particular strain they're able to produce based on using the lactose to produce insulin. Bear in mind, that's not only the limit of metabolic engineering. The idea of metabolic engineering is as well, you can turn a biowaste, right? Whereby nowadays there's a lot of studies being used, even in our UPN, they convert a biomass product, like a waste biomass product, and they turn it into valuable products. These are the things that you can look, whereby you can manipulate your certain pathways within the microbe itself, that they're able to break down and consume this biomass, which turn all these cellulase into a simple sugars, and use these simple sugars to produce insulin. But bear in mind, insulin can also be increased at certain yield, but you have to understand how actually insulin is being produced. You have to know its precursors. You have to know its regulatory. You have to know its promoter. Does the promoter working at maximum level? You have to understand the regulatory. You have to understand its maximum capable value. Sometimes the insulin being produced is actually being subjected to a certain feedback inhibition, meaning that the cell can only produce up to a certain extent. This thing can be bypassed if you knock out or you mute a certain, what I call that, a mechanism or feedback inhibition genes, so that the micro itself can produce the insulin production as much more. So there's a lot of strategies, but yes, of course, in terms of, if you want to, in terms of detailed advice, maybe you can discuss after this. Maybe I can help you to look into this project of insulin to increase the yield at much better rate.
Okay, can I add just a few seconds, Rani?
Yes, of course, please do.
So, yeah, actually, yeah, my problem is a bit different direction, because in my research area, we have a increased insulin signaling that we don't want to, because it's
signaling.
So talking about insulin signaling, but never mind. It's very enlightening. I will talk to you later. So probably we can give other persons the opportunity. Thank you very much.
Thank you very much. Apologies if you don't answer, if I don't answer questions
no
correctly.
No worries. Thank you very much.
Thank you very much.
Thank you. Untuk para peserta, diingatkan kembali untuk dapat mengisi link kehadiran yang sudah diberikan di kolom chat. Now we are going to move to a question which has been delivered into the chat column. Okay. There's one question from Aditya Aryandi, Gajah Mada University, to Dr. Riza. Since the drawback when applying CRISPR-Cas9 into palm case, such as, let me share. Okay, this is the question from Aditya Aryandi to Dr. Riza. Can you see it? Can you read it, Dr. Riza?
Yes, I already read the question.
Okay.
Okay, I can answer it directly. Yes, thank you very much, Pak Adit, for the questions. I think he knows what he's asking. Yes, because in oil palm and other plant crops, you have this problem of off targets, long time consuming, cross-checking, because you need the plants to grow at least 1 or 2 years to be able to check the results. And how to overcome this problem? For now, we don't have any choice if we talk about oil palm. In oil palm, there is not yet in the world even, with many scientists doing a protoplasm system. Why? Because if we have this protoplasm system, we can be, it could be easier to put the CRISPR and Cas9 plasmid inside the plant material, and it can be more easily to modulate, as well as it can be easily to answer the question of the people, the many society that are against the GMO. Why? I can explain it briefly, because you do a CRISPR, right? The CRISPR and Cas9 is not regulated under the Cartagena protocol, means that it is not GMO. The problem is you use agrobacterium in plants. Agrobacterium is transgenic, is the old generation, the old technology. But you combine the two of it, you make something not GMO, put into something GMO, and then you put into the callus and becomes of GMO. But it is debatable. Some people say the agrobacterium did not consist any GMO changes, but eventually when you put it in the cell, you put some of the part of the agrobacterium, right? That is not belong to the plant. That's why we call it transgenic. So in the future, if we can possibly make protoplasm for oil palm, it could be better. But for now, we don't have any choice. So how to eradicate the possibility of the transgenesis? That is why we are using and try to selecting the callus, maybe the second, the third generation of the callus after this one, because we need 1 or 2 years to generate the somatic embryogenesis. Maybe the second and the third generation, we will have only the traits, but not the agrobacterium left over. That is the one of the solutions. And if you're asking about how we apply other beneficial system to produce oil palm rather than creating the tree, we don't have not yet think about that, because right now we have this whole tree of this oil palm that is more efficient of producing oil rather than the others competing species such as canola and the sunflower and the others. So that's why we are going to this direction of producing planting materials. Why? Because the demands are very high right now, and we haven't yet concluded with the possibility of doing a new CRISPR and plant trees in oil palm also. And if you maybe inform about this, the breeding for oil palm takes about 20 years. If we use CRISPR and Cas9, we can cut it until only 5 to 10 years. So at least you will have 2 research scientists that can work together rather than one scientist working 20 years just to do a breeding of oil palm, and then he or she retired having only one varieties of oil palm. But that is the case in plants. In humans and also in animals, you don't have that kind of difficulties. I hope I answered the questions of Pak Adit. Thank you, Pak Adit.
Thank you, Dr. Riza. There is another question for you from Afrida, STT Migas Balikpapan. She's asking about how's the result after editing genes of Ganoderma boninens to oil palm. Was it already trial in field or just at lab?
Yeah, it is already in the field, but in a small, what we call small trial in the nursery. We are waiting the age of the plants. So after we have reached the certain age of the plant, 2 or 3 years, we will, sorry, not 2 or 3 years. So now actually this height for 1 year after acclimatization, we will try to check in the small polybags and try to create small experiments using a Ganoderma just to find out if the editing of the genome are successful or not.
I'm sorry, Ms. Rani, can I talk directly to Dr. Riza?
Oh, sure.
Oke.
Okay. Go on.
Selamat siang, Pak Riza.
Selamat siang, Bu.
Ya, perkenalkan saya Afrida dari STT Migas Balikpapan.
Ya.
Saya pernah bekerja di salah satu universitas, salah satu pabrik kertas di Pekanbaru, kebetulan di RAPP.
Ya.
Jadi kita memang masalah di Ganoderma, baik di HTI maupun di oil palm, ya.
Ya, ya.
Jadi kalau di HTI kebetulan saya menangani HTI waktu itu karena akasia
Ya.
dan eukaliptus. Jadi kalau memang berhasil penelitian Bapak di oil palm, tentu saja akan bisa digunakan research ini untuk di akasia dan eukaliptus.
Ya.
Seperti itu. Jadi memang Ganoderma ini adalah yang paling mengganggu hampir semua tanaman HTI seperti itu. Nah, kebetulan waktu itu saya itu mencari jamur untuk melawan jamur Ganoderma seperti itu. Jamur dilawan jamur.
Ya.
Nah, tapi kalau ada seperti ini, ada editing genes, tentu saja sangat menarik sekali. Tapi seperti saya dengar ternyata biayanya sangat mahal sekali seperti itu, ya Pak ya.
Ya.
Jadi, jadi Bapak hanya baru sampai di lab saja berarti, ya Pak ya?
Ya, untuk saat ini kita sampai di, ya, kita sedang tanam di nursery, supaya nanti tumbuh agak lebih besar sedikit, baru nanti diuji di lapang. Nah,
Bapak sudah tambahkan dalam artian di polybag itu sudah ada ini apa ganodermanya?
Belum.
Sudah ditambah? Belum juga. Ya. Oke. Sebenarnya yang feel yang apa namanya yang yang diserang oleh ganoderma itu biasanya kalau dia second round atau third round. Kalau yang pertama dia aman saja.
Ya. Nah, itu dahulu. Yeah. Sekarang ternyata tanaman generasi baru pun diserang oleh ganoderma karena memang mungkin ada adaptasi dari ganodermanya selama beberapa puluh tahun terakhir. Kan itu tanaman first round berarti sudah ditanam kira-kira 20 tahun yang lalu. Ternyata sekarang ini di tanaman-tanaman baru itu muncul dan bisa terserang juga. Dan gejalanya
dekat dengan ini, fieldnya dekat dengan yang sebelumnya, yang mungkin yang sudah ditanam sebelumnya, gitu.
Ya, karena problematika ganoderma ini sangat sulit karena dia sebetulnya ada di tanah itu bisa berpuluh-puluh tahun dan kita tidak tahu gitu.
Betul.
Begitu lahannya sakit, 1-2 pohon saja sakit, kita bisa bilang bahwasanya lahannya sakit. Jadi hampir seluruh lahan itu yang menyulitkan sebetulnya. Dan yang belum kita ketahui adalah kenapa ganoderma ini menimbulkan case atau problematika di beberapa tanaman saja, tanda kutip ya, terutama tanaman-tanaman yang monoculture atau tanaman-tanaman monokotil. Sebenarnya di kelapa ada, tapi kelapa kan tanamannya tidak semahal kelapa sawit. Itu sebabnya kelapa ya sudah ditebang lalu lahannya dibersihkan dengan dekomposer, dengan pupuk dan lain sebagainya supaya biofungisida supaya selesai begitu terus tanam lagi. Tapi di kelapa sawit tidak, kasusnya tidak demikian. Ternyata sangat sulit sekali mengeradikasi itu. Kemudian, dan mungkin menurut saya juga tidak mungkin juga mengeradikasi ganoderma. Kita mungkin hanya bisa menekan. Itu sebabnya strategi kami ngarahnya ke tanaman. Jadi daripada kita berpikir lahan akan sulit dan bukan ekspertis kami juga, kami berpikirnya tanamannya diperkuat gitu. Nah, kasus ini sebenarnya bisa dibawa ke semua tanaman HTI yang punya problem dengan ganoderma seperti eukaliptus dan lain-lain. Hanya masalahnya memang riset ini mahal itu dalam artian kalau biaya riset mungkin bisa dicari, Bu. Tapi yang sukar dicari adalah ekspertis. Ekspertis, kemudian pengalaman itu kami sendiri kan belajarnya dari nol. Jatuh bangunnya, dulu di awal-awal jatuh bangunnya luar biasa. Karena riset ini baru di tanaman perkebunan dan memang kami ditugaskan untuk melaksanakan itu. Saya pernah mencoba juga untuk bersama rekan-rekan lain di bidang medis, ya ternyata relatif lebih mudah, cuman ada itu plus minus. Kalau di tanaman mau sampai jadi tanamannya tidak masalah, sementara di medis ini begitu keluar sel atau apa langsung menjadi permasalahan besar karena ada problem bioethics. Jadi mungkin CRISPR untuk saat ini ya, menurut saya untuk saat ini di ranah medis pun masih ada di tingkat yang research ya, meskipun beberapa industri di dunia sudah mulai mengarah ke terapi. Kita tidak tahu apakah kedepannya mungkin regulasinya berubah. Nah, untuk tanaman sendiri di Indonesia memang baru dicoba di kelapa sawit, kemudian ada teman-teman di Lembaga Ilmu Pengetahuan Indonesia mencoba di padi dan sudah berhasil. Meskipun efeknya kita belum tahu karena memang risetnya kan baru dimulai 5 tahunan ya. Nanti untuk mengetahui efeknya terhadap traits atau turunan dari sifat itu mungkin perlu sekitar 2 hingga 3 tahun lagi ke depan. Kemudian di IPB digunakan untuk bakteria malah. Jadi sistem dari bakteri dikembalikan ke bakteri untuk melihat apakah dalam fermentasinya dia berubah, tidak berubah, apakah menjadi lebih enhance sistem imunnya dan lain sebagainya. Itu yang saya tahu. Ada beberapa perusahaan swasta juga yang sedang melakukan itu, tapi sampai saat ini saya belum mendengar kabar lebih lanjutnya.
Terima kasih Dr. Riza, juga Bu Afrida. Kita boleh pindah ke pertanyaan selanjutnya dulu ya. Mungkin nanti akan bisa lanjut di pertanyaan lebih spesifik email atau media lain. Oke, saya share pertanyaan dari kolom chat. This is the question for Dr. Riza also. CRISPR is very potential to be used in medical field. You have mentioned about CRISPR-Cas13. Is it possible to be used as a detection system for viruses? Dr. Riza, would you please answer this question?
Okay. I will try to share something also.
Okay.
I'm prepared for this question. So thank you for the questions. I think Miss or Mrs. Denti Rizky Kinanti for the questions. Were very good questions. I was talking also about COVID-19 and CRISPR a few months ago. And now, in general, we have these two technology of CRISPR. We call it detector from Jennifer Doudna, the competitor. And we have Sherlock and Stop from Feng Zhang. This is an industry also. But they created both the same approach using a CRISPR-Cas12a and also 13a. Especially for Sherlock or Stop used by Feng Zhang, they use CRISPR and Cas13. They published several papers. If you're interested with it, you can just Google it, or if you want to know, I can send you. You can contact me by emails, and I can send you the papers about this Sherlock and Stop and also detector. And it was working very well. It was working very well. And they're now trying to go to the more, they try to use it in hospital as like a detection system other than PCR. Because as you know, in this system of CRISPR-Cas used for the detection of COVID-19, what we call, I call it like this, we have more sensible way of detecting the virus rather than RT-qPCR. If I can make an analogy of this, in RT-qPCR, you will need a virus as the size of the Earth, but using CRISPR and Cas base, you only need in the size of maybe 1% of your moon, of our moon. So quite slow. We call it this the concentration or sensitivity in the level of zeta molar. This one, very small amounts, and it was successful right then in the laboratory level, and they have claimed also to be successful in the field, meaning that working on the daily test of COVID-19 in New York in several hospitals. And this one, the technology that we call Sherlock or Stop, has been approved by the FDA, the only one. The detector is not yet. I think they still develop it in the laboratory. So in the near future, I think in the US, they already have this technology. But in Indonesia, if you want to develop this, you will have to start from different angle, maybe different genes to be targeted. And also you have to work with the patent system. Maybe it's quite expensive for us. But if this answers the questions of yours, yes, this CRISPR and Cas13 has been working so well to be used as the detection system of COVID-19. I hope I answered the question. Thank you.
Thank you, Dr. Riza. The next question is from for Dr. Hanan from Bodhi Dharma Universitas Unmul. When you did genes editing, perhaps in fungi or algae, have you got some problems such as off-target editing by CRISPR-Cas9? And how many years you need to finish a project in genome editing to produce a product? Dr. Hanan?
Okay, in terms of off-target for eukaryote, our team have not involved in the modification in yeast or fungal, most of our research is done in bacterial cell, more particularly in Lactococcus lactis or LAB. So yes, if you want to say in terms of even the simplest microorganisms, off-target can always happen. Though, if you move on for simpler microorganisms to much higher microorganisms, from bacterial cells towards to fungal and yeast cells, the off-target gonna be the occurrence of off-target gonna be much higher as you move on to much complex. Their DNA, their chromosomes is much more complex and much more rigid or closely related to each other's. Of course, even in microbes and bacteria, off-target can always happen in any design of CRISPR-Cas9. So not only restricted to any type of fungal, but I would say maybe Dr. Riza can correct me if I'm wrong. As you move on to much higher organisms, it's going to be much more complex, and the occurrence of off-target is much higher. That's the reason why my research mostly at the time is only focusing on bacteria, because it's much more simpler organisms and modern organisms that we're trying to focus.
Thank you, Dr. Hanan. Dr. Riza, this is a new question for you.
Yes. Dan satunya, mereka mengembangkan sistem untuk mengatasi penyakit terkait darah ini, terutama thalassemia, beta thalassemia mayor ya, yang menjadi masalah. Jadi kan permasalahannya yang di beta thalassemia itu adalah penderitanya tidak bisa menghasilkan gamma, kalau tidak salah gamma hemoglobin ya, kalau tidak salah, tidak bisa menghasilkan satu tipe dari hemoglobin itu yang membuat ini mekanisme tubuhnya normal. Nah, mereka berusaha untuk mengkonvert, jadi CRISPR-nya itu mencoba untuk mengaktifasi satu gen sehingga si sel atau ini yang tadinya harusnya menghasilkan alfa begitu, dia diubah menjadi gamma supaya dia sistemnya recompensate. Jadi diubah yang alfa menjadi gamma itu sehingga dia bisa mengikat oksigen kembali, kemudian tubuhnya bisa kembali normal, dan itu sudah dibuktikan di tahap klinis yang kedua. Karena risetnya sudah dimulai pada tahun 2016. Nah, sekarang di tahun 2020 ini kayaknya mereka sudah masuk ke klinis 3 dan mungkin harusnya sudah hampir selesai. Kalau nanti selesai, teknologinya kemungkinan kalau berhasil bisa dipakai untuk terapi itu. Tapi saya tidak tahu berapa harga dari terapi ini, karena mungkin karena ini baru di dunia, itu sebabnya mereka juga mengembangkan ini, Jennifer Doudna, Emmanuel Sarpongki, dan juga Feng Zhang, karena mereka melihat potensi yang besar, satu di industrinya, kedua di permasalahan penyakitnya yang memang selama ini kita belum bisa atasi. Dan jawabannya ya, berarti teknologinya itu memang sudah bagus sekali untuk bisa diaplikasikan di penyakit-penyakit di manusia. Cuma permasalahannya mereka sih belum mengatur ini ya terkait bioethics. Nanti bagaimana, apakah ini menjadi satu hal yang common atau tidak, tapi karena mereka tidak menyentuh germline, mereka tidak berusaha mencoba mengkreasikan atau develop baby CRISPR, jadi kayaknya peraturan ini riset terkait dengan penyakit-penyakit yang yang perlu diatasi di dunia ini pasti diizinkan gitu. Mungkin kalau tiba-tiba ini editnya pada germline gitu menghasilkan bayi yang seperti Hua Jiankui itu menghasilkan bayi yang tidak bisa punya reseptor HIV, atau merubah wajah bayi, atau membuat bayi itu tidak bisa memproduksi protein yang tinggi sekali dan lain sebagainya, mungkin akan menjadi permasalahan yang berkembang ke depan. Mudah-mudahan menjawab. Terima kasih Mbak Rani.
Terima kasih Dr. Riza. The next question is for Dr. Hanan. This is from Bagas, Institut Teknologi Bandung. The question is there any commercial CRISPR-based genome edited product?
Okay, thank you for your question. I think I've shared this. Hold on. The examples of okay, this one. So these are some of the example of what CRISPR have done that has been commercialized. One of it is the bacteriocin which people have done it and tried it before. They use CRISPR-Cas9 to increase a certain genes. They're able to increase the production of nisins. But what we're doing is slightly different from what they have doing. Yes, nisin is one of the commercial products, and other type of products that's been highlighted here that's being produced commercially.
Thank you, Dr. Hanan. The next question is for Dr. Riza. This is from Ahmad dari Malang. This is an interesting method to make a new variety of organism, especially in plants. I just want to know about your opinion. Is that possible if our genetic modified organism can demolish the wild type species in nature? Because most of human more interest to cultivate the good variety compared than wild type with the products with high quality and quantity. So how to tackle this thing? Do you get the point, Dr. Riza?
Yeah, I get the point. Yeah, because this was the question of many debates. So I will try to answer neutrally, because as a scientist, maybe we are working towards the benefits of humans. It is normal. It is normal. But however, we know that what we must understand about the question from Ahmad Rudiansyah is that the changes of genetic drifts, if you want to imagine, it's not only what happened in the laboratory. I think Dr. Hanan will agree with this. It's not only what happened in the laboratory. In nature, it happens naturally, actually, but we don't see it. Because we don't see it, we think that everything that are in nature is normal, is natural. It's not human-made. We take only, for example, we have discussed this with many scientists, and sometimes if you want to have a rough number, I think what we take out from the nature into the laboratory and we try to edit, we try to make a genetic modification is only less than 10% of all the changes that happen in the nature. For example, I will take an example, easy one example. If you have you been gone to Bogor, maybe in Puncak, you will have this kind of manioc. We call it ubi cilembu, right? Ubi cilembu is so sweet. They have a greater taste of honey, but nobody think that it was GMO actually, naturally. Why? Because the sugar itself, it comes from agrobacterium in nature, in the soil, that comes inside the ubi cilembu plants, and they insert genes inside some of the part of starts producing enzyme, and then they produce more sweeter and more likable to human taste. That's why ubi cilembu is quite expensive, and then but it happens naturally. But we never ask this question, right? We never ask where does the ubi cilembu come from? This is only one example, but other examples you have so many. Changes always happen. Likewise, when we see virus right now in the COVID-19, you see daily changes of mutations. It is normal. Why we are so confused right now about this? Because now we see, like a virus, we see the changes, the mutation, the modifications. In higher plant, when you move toward to higher plants, you have more complex regulation and structure of genome. You do not see it because it's quite difficult to see. You can only see human changes for now. We are Homo sapiens. Maybe in 1 billion years ahead of us, we will see changes of us. We maybe have a bigger head because we think too much and etc. But now we cannot just decide that now we change so many things in the laboratory and we did not leave anything in the nature. But nature itself, it's for me, this is my personal opinion. Nature itself is like a moving changes, because we change with the nature. We think that the nature itself is not changing, but actually we change with the nature. So that is why this discussion about the GMO with the conservationists is always going to have more and more theme about this. But do not worry, some of the higher organism did not change so much, because the changes can be corrected by the organism itself. But if you see the lower organism like bacteria, virus, they change so much. And actually, because before COVID-19, we never think about this, about the change in the virus, change in the mutation of a bacteria, for example. We think that there was no change, but actually change is a natural thing that happens around us. I hope that I answered the question. Thank you.
Thank you, Dr. Riza, and also Dr. Hanan. Due to the limited time, we have to end this question and answer session. The rest question will be given to the speakers, and the answer will be sent to participants through email. From this session, I can conclude that genetic engineering and CRISPR-mediated genome editing has a high potential to be applied to not only to microbes, but also to edit plant, animal, and humans. Thank you very much for joining us. Untuk peserta, kembali diingatkan jangan lupa mengisi link pendaftaran agar dapat dikorelasikan dengan pengiriman sertifikat. Lalu dimohon untuk tidak meninggalkan Zoom meeting ini karena acara akan dilanjutkan untuk penutupan dan foto bersama. Mr. Rahmat, thank you for giving us the time, and the time is now yours. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Rani, and thank you to the two amazing speakers, Dr. Riza and Dr. Hanan. Now, ladies and gentlemen, as the discussion session has ended, we are approaching the end of our seminar. But beforehand, let's hear remarks from the chairperson of the Indonesian Society of Biochemistry and Molecular Biology, to Dr. Emma Ridwan. Time is yours.
Sudah bisa didengar suara saya, Rahmat?
Sudah, Bu.
Sudah,
Terima kasih. Terima kasih. I'm sorry for the delay in my welcome speech actually, but now become the closing speech. So due to technical difficulties in the setting, so allow me to deliver my speech in Indonesia to make me more close to you, all of you there, okay? Assalamualaikum warahmatullahi wabarakatuh. Yang saya hormati Ketua PBBMI Cabang Pekanbaru, Mas Dimas, Ketua Departemen Biokimia Fakultas Kedokteran UNRI, Profesor Titania Nugroho, para pembicara tadi Dr. Riza Arif Putranto DEA, dan Dr. Hanan Hasan PhD, seluruh peserta webinar CRISPR in Genome Technology, dan para hadirin yang berbahagia. Syukur alhamdulillah hari ini kita berkumpul di majelis ini dan sudah menerima suatu transfer ilmu yang sangat-sangat bermanfaat dalam bentuk seminar tentang CRISPR in Genomic Technology yang telah dibicarakan oleh dua pembicara kunci tadi. Acara ini diselenggarakan oleh PBBMI Cabang Pekanbaru. Pada pertemuan ini terpaksa saya ganti nih ya, tadinya saya katakan akan dibahas, tetapi sudah dibahas tentang CRISPR, yaitu Clustered Regularly Interspaced Short Palindromic Repeat, satu sequence DNA yang dijumpai pada genom prokariotik yang berperan kebetulan dalam sistem pertahanan antivirus prokariota tersebut melalui mekanisme enzimatik. Misalnya seperti yang telah dijabarkan oleh Dr. Hanan tadi tentang CRISPR Cas9. Mekanisme ini dipakai sebagai tool untuk memodifikasi gen-gen yang perlu perbaikan, sehingga informasi ini nanti akan sangat berguna kepada penelitian kita. Teknologi ini akan sangat bermanfaat terutama di dunia kesehatan maupun di dunia pertanian. Salah satu tujuan organisasi Perhimpunan Biokimia dan Biologi Molekuler Indonesia adalah memajukan dan mengembangkan ilmu biokimia dan biologi molekuler dalam arti yang seluas-luasnya. Antara lain dengan mengadakan pertemuan-pertemuan dalam bentuk seminar, simposium, pelatihan, dan lain-lain. Hal ini ditujukan untuk meningkatkan kualitas anggota yang bisa saya sampaikan saat ini sudah hampir 700 ahli biokimia dan biologi molekuler Indonesia kita yang terdaftar di PBBMI, ya, dan pada akhirnya nanti akan meningkatkan pula kualitas organisasi ilmiah ini. Saya mengucapkan selamat kepada Anda yang ikut hari ini karena dengan menambah ilmu yang telah disampaikan oleh dua pembicara tadi, ilmu yang sangat relevan dengan keadaan kita pada saat ini, yang umumnya adalah anggota PBBMI dari berbagai cabang maupun dari Pekanbaru sendiri. Mudah-mudahan acara ini bisa menambah keanggotaan PBBMI selanjutnya. Dan semoga apa yang diperoleh pada hari ini dapat memberikan manfaat bagi kita semua. Saya juga sangat menghargai cabang Pekanbaru ya. Ini saya pujikan ya. Karena selalu sangat aktif meningkatkan kualitas anggotanya, ya. Jadi anggota cabang Pekanbaru tidak harus mengeluh ya terhadap pengurus cabang Pekanbaru, karena cabang Pekanbaru ini salah satu cabang yang paling rapi manajemennya dan juga paling rajin menambah ilmu anggota-anggotanya, sehingga Anda menjadi anggota tidak rugi di sini ya. Tidak rugi Anda membayar iuran untuk itu semua. Mendapat ilmu-ilmu yang sangat maju seperti ini. Seperti diceritakan tadi oleh Dr. Hanan ya, dengan berbagai aplikasinya, sehingga suatu saat kita bisa mengharapkan dengan sekuens DNA ini kita mungkin bisa menggunting-gunting DNA kita yang rusak, mengganti dengan DNA yang baru, apapun yang kita inginkan. Walaupun masih ada embel-embelnya, teknologi ini mahal dan lain-lain dan sebagainya. Tetapi di mana ada kemauan, di situ ada jalan. Itu yang akan saya sampaikan kepada anak-anakku yang muda-muda, Rani, dan lain-lain ya. Ini sudah ada dua expertnya yang menyampaikan ini. Ayo semua berlomba-lomba yang berminat untuk mengetahui ilmu ini, menghubungi kedua pembicara, mungkin bisa menambah ilmu lebih kalau yang sudah S2, lanjut S3. Yang S3 nanti bisa mengaplikasikan ilmunya di Indonesia. Semoga pertemuan hari ini bermanfaat bagi kita semua, yang jelas pasti akan bermanfaat. Terima kasih kepada cabang Pekanbaru, kepada panitia, kepada Dr. Riza dan Dr. Hanan yang telah membagi ilmunya. Ya, mudah-mudahan manfaat ini akan dipergunakan oleh banyak generasi muda kita yang akan datang. Dan mudah-mudahan pertemuan kita hari ini diberkati oleh Allah Subhanahu wa Ta'ala. Sekian, wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih. Thank you. Sorry. Terima kasih, Dr. Emma. Thank you for the speech, the closing speech. That was supposed to be the opening speech.
Yes.
Ladies and gentlemen, participants, I would like to remind you to fill the attendance form that has been distributed on the chat room, so we can distribute your certificates to your email that will be recorded via the link. And yeah, I would like to share it again. Let me. Okay, it's already been on the chat, and unfortunately, as it is recorded on the chat, as soon as you leave the meeting, you will not be able to fill the link afterwards. So please make sure that you have filled the attendance list before you leave the meeting. Thank you. Before we finish it for good, I would like to invite you all, both guests, speakers, and participants, to turn on your video for a group photo session. I would like to ask help from the IT department and also maybe several members of the Pekanbaru chapter to take pictures. Usually Dr. Arfianti is at hand taking our pictures. You ready, Dr. Arfianti?
Ya, kita tunggu nih, beberapa masih mencoba menyalakan videonya.
I think my
Dr. Hanan, thank you for your presentation today.
Okay. Are we ready?
Oke, silakan dipimpin Mas Rahmat.
1, 2, 3. Yeah, I can surely hear the shutter click. Another time, we already have like 5 sheets of the participants. So yeah, it will take a little bit more time. So 1, 2, 3.
Okay, good.
Yeah, okay.
One more time. 3 is the charm.
Okay,
Third
but
time is the charm.
no one turned on the video. I think no need to take a photo for the third and.
We're already good.
Yes,
Thank you.
good. Okay, thank you, Rahmat.
Okay. Thank you so much, Dr. Arfianti,
kasih, Bapak Ibu semuanya.
our department. And that's it, our seminar this morning. We already approached the midday. And I would like to remind you to fill the registration form to your respective PBBMI branch. If you're already in Riau, please do register for the PBBMI or ISBMB Pekanbaru chapter. As a member, as PBBMI member, you really get all the perks that you can get either from the national society or even regional and even international ones. You will be able to get certain discounts, rating for seminars, and also you are able to apply for fellowships or awards from other society to attend conferences, for example. And also, most importantly, as this is also recorded and broadcasted on YouTube, please do visit our YouTube channel, like and subscribe the channel FK_Unri_official, as there will be more seminars that can be held in this channel. I think that's all. On behalf of the organizing committee, I would like to once again thank you all for joining us and apologize for any inconveniences.
You're welcome. Thank you very much.
Assalamualaikum warahmatullahi wabarakatuh.
Thank you. Thank you.
Terima kasih, Prof.
Everyone, thank you.
Dr. Rizal.
Thank you.
Well done.
Assalamualaikum.